Vanakkam friends, let me discuss on stress mitigating technologies including microorganisms. The stresses are classified into biotic stress and abiotic stress. The biotic stress is caused by pests and diseases. Abiotic stress have four major groups: light, temperature, water and chemical stresses. In the case of light, there are two stresses, low and high light stresses, as well as in the temperature, two stresses, low and high stresses. Again, the low stress is subdivided into chilling and freezing. Drought and flood are water stresses. Salt, pesticides and fungicide are chemical. If you look into the abiotic stresses in detail, the heat and cold stresses directly influence the top growth of the plant, that is stem and leaf, whereas heavy metals, mineral nutrition imbalance will have direct effect on soil health. As well as salinity, drought and flooding will have direct effect on root system of the plant in the rhizosphere. The secondary stresses like ionic, oxidative, osmotic and mechanical stresses will have signaling sensing in the nucleus of the cell. Because of this, signal transduction pathway is being triggered. So this helps in the synthesis of new proteins, and there are two processes are involved. First one is transcription. This means the replication of DNA into RNA by using RNA polymerase enzymes. The RNAs are produced and they have four types of RNA: mRNA, messenger RNA; tRNA, that is transfer RNA; rRNA, that is ribosome RNA; sRNAs, soluble RNAs. These RNAs that are synthesized in the nucleus is transported to the cytoplasm through nucleopores, where it is attached with the small unit of the ribosomes, and this helps in the synthesis of new proteins. And the role of rRNA is to bring a correct amino acids, and the new proteins are nothing but polypeptide chain of amino. Then next, the drought. The drought is the reduction in leaf water potential due to excess transpiration than water absorption. Deficiency or dearth of water severe enough to check plant growth. And the four types of drought: the meteorological drought, that is based on degree of dryness or rainfall deficit and length of dry period. Hydrological drought will have impact of rainfall deficits on water supply, such as stream flow, reservoir and lake levels, and groundwater table decline. Agriculture drought, that is the impact on agriculture by rainfall deficits, soil water deficits, reduced groundwater and reservoir levels needed for irrigation. The socio-economic drought happens when the supply of some economic goods such as fruits and vegetables, grains and meat is lower than the demand. So socio-economic drought happens when demand for economic goods supply exceeds the supply of these essential goods, which is related to weather-related deficit in water supply. The changes due to drought, and we can discuss many changes: loss of turgidity, disrupted cell expansion, and that leads to inhibition of mitosis and limited cell division, and finally the cell death. And there is a damage to the membrane system of the cell, and that will have the generation of reactive oxygen species, namely hydrogen peroxide, superoxide radicals and hydroxy radicals, that leads to oxidative stresses, which all help in decreased photosynthesis, and because of this there is a reduction in growth, inhibition of reproductive development, and finally a drastic reduction in the yield. Methods to overcome drought: first we have to adjust the time of sowing to harvest crops before drought onset. Either you can go for seed hardening with potassium chloride, dihydrogen phosphate and calcium chloride, or the poorly established plants are thinned out. And you can also recommend the plants that are growing in drought, that is growing up drought tolerant crops, that is called as ephemerals, that is short-lived plants. Succulents and non-succulent plants also advocated, and you can go for drought resistant plant. The mulching of soil is also advocated to minimize evaporation losses. Foliar spray of growth retardants like cycosol and malic hydroside is recommended. Foliar spray of anti-transparents like iolin, phenyl mercuric acid, waxes, silicone oils are also advocated. And the next stress is high moisture stress, that is nothing but flooding or waterlogging. Waterlogging means that creates poor aeration, that is nothing but oxygen deficiency and anaerobic situation in rhizobia. The rice is the tolerant crop for oxygen deficiency, whereas tomato, soybean and sunflower are very sensitive crops. And the changes that are associated with the flood conditions are the physiological changes. Flooding induces stomatal closure that leads to low water flow, results in leaf dehydration and reduced root permeability. The leaf decay and wilting of leaves, and there is a accumulation of toxic substances like acetaldehyde and alcohol, and finally the reduction in endogenous level of GA, gibberellic acid and cytokinin in roots. The management of high moisture stress, the following measures are advocated. You have to provide adequate drainage, spray 500 ppm cycosol to arrest apical dominance and promotes growth of laterals. Foliar spray of 2% diammonium phosphate along with 1% potassium chloride in the form of muriate of potash, or spray of 40 ppm naphthalene acetic acid or 0.5 ppm brasinolide or 100 ppm salicylic acid is also recommended, as well as foliar spray of 0.3 boric acid along with 0.5% zinc sulfate plus 0.5% ferrous sulfate and 1% urea. Nipping of terminal buds in cotton to arrest apical dominance, that will promote growth of symbodial. The next one is temperature stress. There are two stresses, as I told, the low temperature stress and high temperature stress. In the low temperature stress, again two types: chilling injury, when the tropical plants are exposed to 0 degrees Celsius, and another is freezing injury. This occurs at temperature below 0 degrees Celsius. Whereas in the high temperature stress, the heat injury is the major one when plant temperature is higher than environmental temperature, that it exceeds 35 degrees Celsius. The physiological effects of low temperature stresses are outlined here, and the cell membrane is weakened and leaky. That is the first physiological observation. That leads to inactivation of mitochondria, where the respiration takes place, stoppage of protoplasm streaming, and accumulation of respiratory toxic substances, and very important, ice formation in intracellular spaces, and finally growth of plants are arrested, which is because of cell membrane lipids get solidified. What are all the mitigation measures to overcome low temperature stresses? Are seed hardening with 0.01% ammonium molybdate, or foliar spray of 0.1% ammonium molybdate, foliar spray of 2% calcium nitrate, 2% DAP plus 1% KCl, salicylic acid 100 ppm, 500 ppm cycocel and 0.5 ppm brasinol. And what are the physiological effects that takes place during high temperature stress are mainly the oxidative stress that will lead to inhibition of seed germination, reduction of plant growth, improper development of the growth, alteration of photosynthesis, alteration in phenology, that is nothing but morphology, alteration in dry matter partitioning, water loss, and finally yield reduction that also results in the reduction of crop quality. The mitigation measures for high temperature stresses are seed hardening with 0.5% calcium chloride, foliar spray of 2% DAP plus 1% KCl or 40 ppm naphthalene acetic acid or 0.5% brasinolide or 100 ppm salicylic acid, or otherwise you can go for foliar spray of 3% kaolin. In addition, we can go for foliar spray of 0.5% zinc sulfate plus 0.3% boric acid plus 0.5% ferrous sulfate plus 1% urea. And you can also advocate foliar spray of PPFM, pigmented facultative methanotropes at 10 to the power of 6 as a source of cytokinin. The question and answer: the upward swelling of soil due to frost is called as, the answer is heaving. The underflood condition, the deficiency of, the answer is oxygen deficiency occurs. The chemical used for seed hardening to overcome drought is potassium dihydrogen phosphate. That is the correct answer. Example of anti-transparents are kaolin, waxes, phenyl, mercury chloride. These all the answers are correct. So the option for this question is D, all the above. Good friends, we have so far seen the two stresses, light and temperature stresses, the salt stresses. The salt stress is due to excess salt accumulation that results in decreasing in osmotic potential, which resulted in X-osmosis and wilting of plants. There are two types of salt stress, the saline soil and alkaline or sodic soil. Saline soil, the electrical conductivity is more than 4 dsm to the power of -1. The ESP is less than 15%. pH is less than 8.5%. The saline soil is mostly dominated by chlorine and sodium. The alkaline or sodic soil, the electrical conductivity is less than 4 dsm to the power of -1. The exchangeable sodium percentage ESP is more than 15%. And the pH is more than 8.5, and this soil is dominated with carbonate and bicarbonate. The physiological effects of salt stress that is being explained here in detail. The first physiological effect is delays in seed germination, reduction in seedling growth that will have reduced vegetative growth, and all these growth development affects panicle initiation and spikelet formation, and also the higher uptake of salt and high transpiration rate. These growth and the salt accumulation effect will have direct effect on fertilization and pollen germination that declines photosynthesis, and finally there is a reduction in chlorophyll. Based on salt stress tolerance, the plant kingdom is classified into 4 types. The halophytes, the plants that grow under salt stress concentration, is called halophytes, and the plants that cannot grow under high salt concentrations are glycophytes. The plants that can tolerate moderate salt stress is oligohalophytes. The plants that can tolerate extreme salt stresses are euhalophytes. The highly salt tolerant crops are, the examples are cotton, sugar beet, asparagus, date palm and barley. Moderately tolerant crops are rice, sorghum, wheat, safflower, soybean. And moderately sensitive crops are rice, maize, cowpea, groundnut, sugarcane, tomato, potato, radish and cabbage. The Crops that are extremely sensitive are citrus, strawberry, melons, peas, carrot, onion. The salt stresses can be mitigated by seed hardening with sodium chloride 10 molar concentration or incorporation of danger at the rate of 6.25 ton per hectare, split application of N and K fertilizers along with gypsum, foliar spray of pink pigmented facultative methanotropes at 10 to the power of 6 as source of cytokinin, foliar spray 2% diammonium phosphate plus 1% KCl or 0.5 ppm brasinolide or 100 ppm salicylic acid or 40 ppm. The microorganism in stress management. This is very important. The microorganism plays an important role in defense mechanism against any stresses that is known as induced systemic tolerance, in short IST. Microorganisms also alleviate abiotic stresses called, and these microorganisms are called plant growth promoting bacteria. The best examples are Pseudomonas species, Bacillus species. And how the abiotic stresses are managed by the beneficial microbes that is explained here in detail in this image. When you apply the beneficial microbes that will produce the jasmonic acid, ethylene, pathogenesis and related proteins that will have direct effect on induced systemic resistance. Similarly, the biotic stresses are also managed by pathogens like virulent, avirulent, non-pathogenic microbes and chemicals. These all produce the salicylic acid and pathogenesis related proteins that will have systematic occurred. The mechanism of action of different microorganisms. If you apply bacterium, that will produce phytohormones like auxins, indole acetic acid, cytokinins, gibberellins, and phytoalexins, along with plant growth promoting substances that will help in solubilization of phosphates and potassium, as well as nitrogen fixation that also enhances the uptake of macro and micronutrients. If you apply the fungal pathogens, that will produce enzymes, antibiotics, heterophores, and osmolytes that will have direct effect on improving the texture and structure. And if you apply the beneficial microbes that will secrete organic acids to decrease soil pH, and this will help in solubilization of inorganic and insoluble phosphate into soluble phosphate. The microorganisms involved in this process are Azospirillum, Astrofactor, Bacillus, Penicillium, Pseudomonas, and in this diagram, how the abiotic stresses like cold, drought, heat, heavy metals and salt stresses are affecting the plant kingdom. I can list out one by one. The excess reactive oxygen species accumulation that will have a membrane dysfunction and nutritional disorder, hormonal imbalance that also reduces the photosynthesis and protein synthesis. When you apply the beneficial microorganisms that will synthesize the cytokinin, gibberellic acid, indole acetic acid, salicylic acid and abscisic acid in the root zone area of the soil, that will help in improved germination, upregulated antioxidant system, reduced ROS accumulation and induction of system acquired resistance, and finally assist in the cell signal. The last part of this presentation is the different varieties that are tolerant to different abiotic stresses like drought, high temperature, salinity, flood and submergence, low temperature and rise are furnished in this slide. In the next slide, the pulses varieties like green gram, black gram, cowpea, red gram, Bengal gram, soybean and horse gram are furnished. Similarly, the tolerant varieties of oil seeds like groundnut, sesame, castor, sunflower and coconut are furnished. The final part is the question and answer. The example of salt tolerant crop is the cotton is the best answer. The dosage for brasinolide used to increase photosynthesis activity under stress condition is 0.5 ppm. The microbe used to overcome drought condition is ppfm. That is the correct answer. Alkaline soils contain more of carbonate and bicarbonate ions. For PDF and online test, kindly text me by using this mobile number. That is also WhatsApp number and my mail. And kindly subscribe my channel. Kindly share, like and comment my presentation. Thank you very much.